Our first speaker is Professor Baptiste Ramond from Pontificia Universidad Católica de Chile, and he will be speaking about functional profiling of Atacama Desert edaphic niches. Thank you, Baptiste, and take it away.
Yeah, hello. So I'm going to put my presentation. Thank you very much for the introduction. Thanking you a lot also to give me the opportunity to present this thing if I can manage it. Sorry about that. Where is it? There it is. There we go. So you see it well? Or should I switch?
It's in the presenter view, so you should switch.
Okay, so I'm going to switch. Dun, dun, dun.
Or duplicate your screen.
Where is this thing? Oh.
It's above where it says parameters d'affiche or something like this in French.
Paramètres, because there's too much stuff there. Deactivation. Ah, that one. No.
There,
Yeah.
that's good.
There's the names. And which one should I take?
You should put it in full screen now.
Okay. Sorry about all that. Be working, hopefully. Is it?
1 second loading.
Okay, well, so I'm going to try to introduce this. So yes, so thank you. So I'm going to talk about some of the research that I've been doing in my lab for the past 3 or 4 years now since I'm in Chile. And we're going to talk about, obviously, one of the emblematic deserts in the world, which is the Atacama. And I'm going to focus on the most extensive niche that you can find in a desert, which are the soils. And so does it work?
No, it still is not in full view, full screen. So you should go.
Let me just stop this to catch. I made it work like just before.
There.
That's perfect.
That's perfect.
Yeah, just
Okay. Sorry about that.
Go to the first slide.
So we're back in the first slide.
You have drylands are the most dominant terrestrial environments.
Okay. There we go. So yeah. Okay. So why it's important to study drylands or deserts is actually because if you look at this map, you can see all the land that is not in gray is actually considered as a dryland. A dryland is defined by its aridity index, which is the ratio of the precipitation over the evapotranspiration. And as you can see, you've got different levels of drylands. And if you take deserts in particular, they have aridity index below 0.5. And yeah, that's the difference. So you've got 4 different regions within the drylands. And another definition that's important to actually acknowledge when we talk about drylands or deserts is actually they're not all the same. Obviously, we've got cold deserts, which have a mean annual temperature over below 18 degrees Celsius, and the opposite, the hot deserts, which have mean annual temperature over 18 degrees Celsius. And then we've got the polar deserts, which are not on this map, which have for a warmest month, the month that has the mean annual temperature, mean monthly temperature below 10 degrees Celsius. So as you can see, the Atacama Desert, which I'm going to talk about, is one of the cold deserts in the world. Deserts are very interesting because they are polyextreme ecosystems. So as you can see here, they are hyper-arid. Well, that means that they have a deficiency in water, which is mainly due by the fact that actually they have very low precipitation. They are oligotrophic system. That means they're very poor in nutrients. They have extreme temperature fluctuation, whether daily or seasonally. They have high incident UV radiation, and in some cases, very high winds or katabatic winds, like in Antarctica, for example. All in all, this has for a result that generally the higher plant and animal life is low and limited. And therefore, as microbial ecologists, we like to say that the microbial communities and deserts are actually the drivers of most, or if not all, the ecosystem function, important ecosystem function, such as primary productivity or nutrient cycling. So Why is it it's important also? So this is actually a figure I took during the lunch break from the BBC website, and we can see that actually, for scientists, we quite know it, that actually we are having a climate shift, a climatic shift. The world is actually heating, and 2023 is now officially the warmest year on the record. Okay, what does it entail for us, or actually generally in deserts, is that we are having a global desertification or land degradation, which is the fact that there is less water entering the system. So that means that a dry subhumid environment is becoming semi-arid, a semi-arid one is becoming arid, and an arid is now turning into a hyper. And what you can see in this figure, which are different photographs I took, is actually, you can see the vegetation cover is actually decreasing, which means that actually productivity in the world globally is actually decreasing, which can be a problem in general, is that as of today, we know that 35% of our population actually thrives or actually live or depend on deserts or dryland for their subsistence, because 44% of all the cultivated systems that you can see here and 50% of our livestock actually live in those regions or the surrounding regions. With desertification, there's a big threat that actually over 250 million people are actually now affected by what is happening nowadays. And we can consider over 70% of the dryland, so 70% of 50% of our surface is actually in a degraded state. And now, to have this a bit microbial footprint or view about what I'm interested in, is actually, this is from a figure actually from an opinion we wrote with colleagues. And if we do not act, we do nothing, actually there's a big problem, because we can actually estimate that today the surface increase of deserts is 23 hectares per minute. It's huge. And with this come a loss of the biodiversity and productivity. Obviously, in the next 40, 30, 40 years, what it means is that 1 billion more humans are at risk of famine and all the consequences of a global desertification, and over 50 million people might be displaced. So this is the pessimistic part, but actually it has to be talked about. And so what we say, what we propose, actually, we can actually use microorganisms or actually desert microbes to as a biotechnology or as a let's say to impede or impeach a bit the desertification process or mitigate the desertification. For example, by cultivating biological soygrass, by improving the resistance of plants to dry condition, etc. Now, what is special about the Atacama? So the Atacama, as you can see on the map on the left, so it's this really narrow region that we have in Chile, the northern Chile, that goes a bit into Peru. And it's actually so over 2,000 or close to 2,000 kilometers. And you can see on this map of Chile, where you have all the different aridity region, that it's roughly this whole, what is called on this map, the xeric plate, the xeric regime. And you can see that if you go inland, it changes a bit. Okay, so it's not the whole desert that you can consider hyper arid. It is, however, the most arid region on Earth if you don't take into account the polar desert. What is interesting is it's one of the oldest, but not the oldest deserts in the world, and it began to be its desertification began around 50 million years ago when the Drake Passage opened, more or less, because it's changed all the currents, and now we've got, for example, a cold upwelling here that actually is shifting all the climates around here, and water is not able to actually penetrate from the sea. And also because we've got the Andes Mountains, also we've got an exclusion from the Amazon rain. And this has led to arid to hyper-arid condition around 15 million years ago, which means that the Atacama Desert is the oldest hyper-arid desert in the world. Obviously, because of this kind of island setup, there's lots of endemism. Like, for example, 60% of all the plants are considered endemic to the Atacama Desert. And just as a very fast mention, there's been lots of studies because it's been a hotspot for astrobiology studies, for example, and microbial communities and lots of its niches are diverse, they're active, and obviously they show very good adaptation to those extreme conditions. So going back to characterizing a bit, so it is the Atacama Desert is actually has for origin both a climatic and a topographic consequence. And what you can see on the map on the left is you've got 5 little regions. So you've got the coastal region around here, which is separated, like we can see here, by the coastal Cordillera. Then we have a central depression that you can see here. Then we've got another mini Cordillera, pre-Cordillera, second depression, and then the Andes per se. What can we say if we look at, if you go there, for example, this is the transect. This transect here is a transect that's around there, like more or less like what in the region that Mauricio showed around here. And if you go to the sea, you've got a lot of fog, but the fog is limited, so it's almost daily, is limited by actually the mountains here. So you've got a zone that is influenced by the fog to the coast. Then in those little depression, you have a very strong and hyper-arid state. As you can see on the picture, there's absolutely no evidence of life, no plants, nothing. Then as Mauricio showed us previously, then we go in the Andes, you've got different ecoregions, I would say, that have different vegetation cover, temperature, etc., as you can see here, and they vary depending on the temperature. However, if you take the whole desert, which goes then from Copiapó, as it's on the map here, to Iquique, depending on where you are, you've got very specific or very unique things that you can see. For example, in the south of the Atacama, when it rains, which is rare, we've got this very famous event, which is the Desierto Florido, where you've got a massive bloom, which is fantastic to see, but it's very localized. If you go in the north, in the region of Iquique, if you go at 3,000 meters, you can see that the Prepuna is not actually the same. You don't have the same vegetation. So knowing all this, we said, okay, we're going to work on the functioning of the Atacama Desert. So try to take into consideration all of this variability. And the hypothesis related is, okay, we've got here a graph of the soil activity with the transect of the desert. And what we lay around, so okay, we've got a zone impacted by fog, a hyper-arid zone in the middle, and what we say a mountain zone that then we can separate in different things. And the hypothesis is that the soil activity would actually be strong in the fog zone because of the amount of water that comes thanks to the fog. It would drastically go down up to maybe 0 because it's, as I said, one of the most hyper-arid, it's the most hyper-arid desert in the world. And then it would increase again in the mountain zone where it would probably be limited at one point by the temperature. So how we did this? So we designed 3 different transects that goes from the sea to the mountains. So we're limited basically by where the roads can go, more or less. And it goes from 0 to 4,800, 900 meters. And we have 17 sites. In all of these sites, we've got 3 individual sites that are far away, like 150 meters to 100. And within these sites, we took 3 true replicates. So we've got this amount of samples at the end. We studied soil enzyme activities. So this is one way we do it. So it's extracellular activities. What we did is, so basically you give a substrate, an analog substrate to an enzyme, and you look at its activity when it's cleaved. So we've got 4 enzymes that we tested, one for carbon acquisition, one for carbon and nitrogen, but mostly nitrogen acquisition, one that is for nitrogen acquisition only, and one for phosphorus acquisition. We also tested over 1 year the carbon degradation potential of these soils by laying down different type of carbon sources, so pure cellulose, tea bags, and Roy's Boys tea, which are classic experiments done in the field at the surface layer and the deep layer. We also measured greenhouse gas production at different sites and started measuring, so this is a preliminary result, chlorophyll A concentrations to have an idea of the primary production potential of those soils. So just going to start with the last I've just presented is because actually this is preliminary because we only have results from these little places and very little replicated. But what is interesting to see is actually we did detect, even though a low amount, chlorophyll in all of the soils, even in the zone where actually we see no plants. So that means that probably in those regions we have chlorophyll from algae, microalgae, and cyanose, and that means that there is a potential primary production, microbial-based primary production. And then we see there's a trend maybe that's going up, but this is fast to be. So then we tested the greenhouse emission consumption. So as you can see here, we've got the carbon dioxide production. Depending on the altitude, you can see the different transects depending on the color. So as you see, they're not all going to the same altitude, etc. But we basically go from the fog zone to the steppe zone. And we have respiration, so measured in 90% of the sites. We clearly show that there is much more respiration happening in the place where there's more vegetation, so probably carbon inputs in the mountain soil, and we do not see any fog effects. We also see a very interesting sharp decrease in the steppe zone, but this is really because we reached the top of the mountain and there's actually no biomass or no vegetation cover. For the methane, sorry?
1 minute.
Okay, so for the methane production, we can see actually consumption, and it's the same. We only see that it's in the mountain region that we have a lot of consumption. In the degradation, we can see also depending on the site. So this is the control. We can see the activities, and the same as you can see. Sorry,
we lost you for
so green tea.
There. We lost the connection for a few seconds.
Okay, sorry. So here's the same type of graph with the green tea, the rooibos, and the cellulose, and you can see the same that on surface and deep, we've got different degradation depending on where we are, and it depends on the substrate. Cellulose is the most degraded compound, and there's no fog effect, while we can actually see a mountain effect. And for the extracellular activities, we can see if we look at all the different enzymes that I talked about, that is active community throughout the transects, almost independently on the enzyme. And even in the hyperarch section, which was a surprise. And we even see that one of the enzyme, which is NAG, which is here, is actually quite active in the hyperarch zone. There is a fog effect, as we can see with the black arrow here for some of the enzyme. And there is actually, like always, like a potential activity hotspot in the mountain regions. Some, well, just to show that we did some MAG reconstruction, and we have very, very new species potentially. These are the best MAGs that we generated, and they all have like very nice, interesting functional capacities. This is actually work in progress. So take-home message. So the Atacama Desert is actually active or potentially active, because some of the measurements, we don't know if we're measuring the soil, what the activity of cells, and it's actually independent more or less of what our zones we defined. Even though we see that the Andean samples appear to be the most active soils. So all in all, there is an effect of the climate and the topography, but something else, and we are measuring now soil chemistry to know. And the take-home message is that as microorganisms from the Atacama Desert soil, which is super extreme, are able to be active, we can actually probably use them to fight desertification and improve dryland agriculture. I would like to thank everybody for your attention. This is the end of the trip with me. I would like to thank particularly Ivo and Natalie, because it's their work I presented. I just have the chance to actually talk, and all my funding bodies and collaborators on the project. Thank you very much.
Thank you very much, Jean-Baptiste.